All right, good afternoon, class. So welcome to biology. We're going to start with some of the calculations needed for microscopes. Now, don't freak out, the math is fairly straightforward and is limited to just this concept in biology with the exception of one small part. We're going to do some math later down the road. Now, you should know by now what the compound microscope looks like and what the different parts are. So as a refresher, we have two different lenses on every microscope. We have the eyepiece lens, also known as the ocular lens. So that's this ocular. And then we have the objective lenses. And the objective lenses are the ones that you can rotate to change the magnification of your specimen. So you see here that there's 10, and this is probably a 4 times, and this is probably 40 times, or something in that area. And you can just rotate that plate here, and you can see the different lenses. In order to get your total magnification that you'd see up in your eye, you actually have to multiply those two magnifications together. So it's your eyepiece times your objective. Your eyepiece in the microscopes we're going to use is always a 10 times magnification. So in order to get your total magnification, it's going to be 10 times whatever magnification you've chosen as your objective lens. So if it was, say, 4 times, then it'd be 10 times 4. So it'd be a total of 40 times magnification. So that would be your example here. For a 4 times magnification, if you look at your formula sheet, it's the power of the ocular lens, which is going to be 10 times. So it'd be 10 multiplied by your power of the objective lens, which is 4, equals 40. If we were to do the 10 times objective lens, then it would be 10 times 10, because that would come from here. This one went here for 100 times. And then the maximum that we can get in our compound microscopes at the school is the 40 power objective lens, so 10 times 40, or 400 times magnification. That would be for this one. Okay, so that one's a super straightforward calculation, but you will need to know how to do that. Okay, the next concept is the field of view. Now, field of view is just what you can see when you put your eye to the microscope. It'll have this circle, and you'll see, I don't know, whatever you're looking at, if you see cells or anything. So your field of view is just what you can see. The value for a field of view is the measure of your view diameter. Now, if you're not so fantastic with math, the diameter is the distance across the entire circle. That's your diameter. That's your field of view. That's just how much you can see. Usually it's measured in millimeters or even micrometers. Some of them, like the really low powered, when it's 40 times magnification, you can actually measure the field of view yourself. And that's what we would do in a lab if we were in a lab. And usually what that'll look like, if you took a little ruler and you put the ruler under the 40 times magnification, it would look something like this. And you'd see little tick marks. And it's usually around 3.5, 4 millimeters. So in this case, if this is in millimeters, this is 1 millimeter. Then you have 1, 2, 3. Maybe I'd say that would be I'll round up and say 3.5 millimeters. So, if I said my field of view diameter is 3.5 millimeters, notice how I used the whole diameter and not just from here to here. It's the diameter is the biggest line you can make in a circle. It cuts the circle in half. So this would be 3.5 millimeter field of view. But let's say I magnified that even more. Well, what I would see would be, I'd have my ruler here, but my ruler would actually be quite big, so I wouldn't even see the bottom. And I would just see something like this. And it'd be a lot harder to measure and these would be bigger. This would be like this, because they'd be massively blown up. So I'm zoomed in more, because I'm 100 times magnification instead of 40. And then, if we looked at 400, all I would see is this big, thick thing, and maybe one line. Maybe. These are just approximations. But obviously it's very difficult to get an accurate representation on how wide this field of view really is. So in order to get the field of view diameters for these two, we need to use math. And the reason we need this is to see how big our specimen really is. If I look at a little cell, it's important to know if I'm looking at it with a certain magnification, but it's also going to be important to know how wide that field of view is so I can guesstimate the size of the specimen. We'll talk about that a little bit later. So let's look at the formula sheet. Our formula sheet has high power field of view is equal to low power field of view, which is what we measure, times the low power magnification divided by the high power magnification. Now I know this is just a lot of words, but it's quite simple when we break it down. And one thing I want to just kind of clarify, it's not high power as in the highest power, it's just the higher of the two that you're looking at. So I'm going to cross those out and say higher and lower. So let's look at an example of one that we've already done. So we've already done this one where we've measured the field of view for the lowest power. And we're going to do the 100 times magnification. So the higher power field of view is the field of view that we're trying to find for this 100 times field magnification. That's going to be equal to the lower power field of view, which is what we measured. So we measured the low power field of view as 3.5 millimeters. Times our lower power magnification, which is going to be 40. Then divided by the higher power magnification, which is the 100. And if I multiply 3.5 times 40 divided by 100, I get 1.4 millimeters. Which, that's about 1.4 millimeters right there. So that was probably a good approximation. Notice how I can see less distance when I magnify my results. That makes sense because you're looking even closer. So you're only going to see a certain width. It doesn't mean you see less or more in terms of detail. It just means that the space that you see, the length of the space that you see, shrinks when you zoom in further and further. Let's do just on the side here. I'm going to do the 400 times magnification to give you a little sense of how little we see, or how narrow of a window we see. So I'm going to make that equal to my 3.5 as well, because again we're going to use what we measured and then the higher power magnification. So 3.5 times the lower power of 40, but this time my higher power is going to be the 400. And that's going to be 0.35 millimeters. So we only see 0.35 millimeters across the whole field. So this here would equal 0.35 millimeters. Alright, let's try a new example. If an objective lens of 4 times has a field of view of 4.5 millimeters, what is the field of view for a 40 times objective lens? Alright, so what that means is this. I'm going to make this a better circle. There we go. For the 4 times lens, this field of view is 4.5 millimeters. Then, for the 40 times, we want to find out what this number is here. But what I need to change down here is the amount that is actually being multiplied, the total magnification, because this is just an objective lens. That doesn't include the ocular lens. So we need to find the magnification for both lenses. And what that's going to be is this 4 times 10 times, because remember the eyepiece lens is 10 times magnification. So this is actually a 40 times combination, the compound microscope. This one here is going to be 40, I should do this, 40 times that 10, which is 400 times. So using a 40 times objective lens actually creates a total magnification of 400 times. Now, plugging in our formula, the higher power field of view is equal to the, I'm going to get rid of this stuff, equal to the lower power field of view, which is this guy here. So 40, oh sorry, not my field of view. My lower power field of view is 4.5 millimeters times my lower power magnification, which is my 40 times, and divided by a higher power magnification of 400. And that is going to give me 0.45 millimeters as my field of view. Awesome. Hopefully that makes sense. If it doesn't, there's some more practice examples in your textbook. Alright, now what's the point of this? Sure, we now know how wide across that is, but what that's going to help us do is estimate the size of the actual objects. So I have here an example of an onion cell. This is an actual onion cell that I have taken a sample of and taken a picture through the microscope. And if I was to ask you how big that is, you would not be able to tell me, because we don't have any information. But if I gave you the field of view, now you can tell me how big it is, because you have some sort of reference point. If the whole onion cell was this big, and my field of view was 4.5 millimeters, you could tell me the onion cell was 4.5 millimeters wide. Now, this one is made up of multiple, but we can use the amount of cells that fit this field of view to solve for how big the onion cell is. So, the formula for object size in your formula sheet is the field of view divided by the number of objects needed to fit across the field. So, let's take that out. If we were to use the same objective lens here and use my field of view, this is in the highest magnification, so this is going to be my field of view of 0.45 millimeters. Object size is equal to field of view 0.45. If it was using the smaller, or the bigger field of view, sorry, then it would be a lot more, it kind of looks something like this. There'd be a lot more onion cells in the field. But now we've zoomed in a lot more, so my field of view is smaller. So 0.45 millimeters divided by the number of objects that fit across the field diameter. So from here to here, we're going to count how many objects. Now it's a little bit harder because they're kind of on an angle. So I'm going to go from here all the way to here. And I count one big one, and then kind of half of another one. So on average, I kind of got to make an average width. I'm going to say that there's a total of 3 cells that go across. And this isn't the object size, this is the object width. And my object width then is 0.45 divided by 3, 0.15 millimeters. Which, if this was a real example, would tell me that my onion cell is 0.15 millimeters wide. Then, if I wanted to find the object height, I would erase this, and I would count the number of objects going this way. And I've got 1, 2, 3, 4, and then a little bit here and a little bit here for 5. So it'd be 0.45 again, divided by 5, which would tell me my onion width. This one would tell me my onion height. And that height would be 0.45 divided by 5. 0.09 millimeters high. Now that's only if these were actual numbers. I just made these numbers up. Okay, so that's what the point of finding field diameter is, to eventually find your object size. If I was to give you this picture here, you would have to find what the scale is. Scale is used to explain how much bigger a picture is than the actual object. The formula for scale is your drawing size divided by your object size. So you'd actually take a ruler and measure the length of this drawing, and then divide it by the actual length, which is your field diameter. So I'm actually going to take a ruler and find out how wide this scale is. This ends up being 6 centimeters wide. So 6 centimeters. And then I'm going to divide it by my field diameter of 0.45 millimeters. Notice how they're in the wrong units? I have to convert centimeters into millimeters. So this is 60 millimeters over 0.45 millimeters. And I end up with 60 divided by 0.45, which is 133.3 divided by 1, or 133 to 1 is my scale. So this tells me that right now this picture is 133 times bigger than the actual object. So this photo of the cell here, this guy, is much, much bigger. Awesome. Well, that's all for now. If you have any questions, please reach out. If not, have a great day.